Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group. I'm really excited to have George Hosu here today, and he will be discussing solving aging by breaking out of ontologies. George, take it away.
Wonderful, thank you. So yeah, I am George. I am actually running a very tiny neuroscience lab right now, together with Asher, trying to do the whole independent scholar thing. Oh, Asher's here. Yeah. Okay, wonderful. Together with Asher, that's in the meeting here. And yeah, I want to talk about this important piece of cognitive technology that I've come to conclude we need in order to solve aging, which is an ability to more fluidly navigate ontologies. Another way to put this is the ability to be more loose with the models we use to make sense of reality. This doesn't mean less rigorous, quite the opposite. I think we should significantly increase our rigor and really ask which models deserve the mind space and the funding that they actually get. On the flip side, I believe we should be more accepting of initiatives that present interesting data, but fit no particular model or no particular ontology. Now that's, sounds quite serious, and partially I'm joking, because what I don't want to be is like another sort of like annoying tech pro guy coming into biology and talking about like meta science and systemic changes. What I do want to do is provide some examples of what it looks like to think outside of existing ontologies and try to encourage people to not be willfully ignorant of observations that don't fit a popular ontology, nor, more importantly, to forego their own interesting findings just because those findings do not fit an ontology. So the one prerequisite I have here is reminding everybody, including myself, this thing that we already know, which is that we shouldn't confuse base reality for scientific theory. In some narrative, you can even think of science as being originally created by people refusing to think inside existing ontologies at the time. Those being primarily these like Aristotelian and Neoplatonist models with various elements from Abrahamic religions mixed in. And instead, they choose to initially look at the world directly, try to use less of ontological filter. And then they started to model how processes of change were happening. Now, as scientific tools became more advanced, we've solidified them into their own ontologies. But it's like really important to just remember that ultimately these ontologies came about because they are useful. It's in some fundamental sense no more meaningful to say that there is such a thing as a particle in the standard model as there is to say that there is an angel on the crystal sphere spinning the sun. It just so happens that particles in the standard model tend to describe reality a lot better. So, let's just remember to never really treat like scientific abstractions as real. I don't want to comment on, what metaphysics you use, but no matter what those are, we should remember that these ontologies, these models, these theories exist to help us make sense of the world and not the other way around. Okay. So with that aside, I would claim that there is currently no good ontology for understanding human aging. I think that if we had one, we would have solved aging by now, or at least we would have put a dent in the problem. We have studied medicine and biology for over 100 years, and this has had great effects on median lifespan. It's had great effects on things like childhood mortality. It probably has had great effects on overall quality of life. But it's had no effect whatsoever on maximum lifespan, and not even in delaying the rate of aging in the people where it's slowest. Not even in humans, even if you're a mouse. Same story. However, there's plenty of interesting findings, and I think some of them might give us useful clues as to how we should go about understanding the process of aging. I think these often get ignored, not in a malicious way. They simply don't fit into anything that allows them to be pursued further. To create something novel right now, you have to make it fit both the scientific language game, and at some point you have to make it fit a commercial paradigm. And this forces novel ideas to often play outside of the spaces that generated them. And the generators of those novel ideas often keep getting ignored because of that. I want to look at some examples that I honestly picked somewhat arbitrarily. Intentionally, I picked examples that I don't put that much weight on, more so to just say, here's some things about the world that are quite interesting, yet that most of us are not able to integrate in our models. And they're somewhat neurobiology focused because that's my hobby horse, and I might as well focus on that. So let's go for these really quickly. So the first is gamma wave stimulation. And this is the kind of thing we could do in the 60s. It's just using flashing LEDs and binaural or monaural beats at a specific frequency, which we often notice when we do EEGs. And recently we've had trial data for this, and this is like tentative. Don't take it as a very definitive claim. It does seem to work better than the two best FDA-approved Alzheimer drugs. And many people have speculated as to why this kind of thing would work. And I think it should tell us something about we need to study Alzheimer's at the connectome level. I'm not saying that necessarily what we need to do is, have gamma wave stimulation to fix Alzheimer's. Actually, I think it's like very unlikely, but it's interesting that this sort of 101 cognitive stimulation paradigm has been able to yield comparative or better results on this disease than hundreds of billions stuck into the small molecule approach. And in large part stuck into this amyloid plaque accumulation model, though not only. Next up, this might be a bit harder to understand, but I do want to talk a bit about neoneurogenesis, because this is one subject I do actually care deeply about. And my one conclusion with it is that nobody has any idea what's going on at the cellular level. I could talk about conflicting studies here, but instead I want to talk about this interesting thing, which is that white matter progenitor cells are likely also neuronal progenitor cells. And I think this is a good example of how an ontology fails to update when we have a very interesting finding. We start with this consensus position that there is negligible or no neurogenesis in the adult human brain. Okay, maybe a bit of it happens in like the olfactory bulb and the hippocampus, but like really there's nothing once you're past the age of 2, even there. And then you have evidence about things that, when extracted correctly, can actually differentiate into neurons all over the brain, including areas that are transitive and don't have a lot of neurons, like white matter tracts. We also know that cultivating neural progenitor cells in vitro is quite hard. They tend to die or differentiate quite quickly. So the fact that we found these like seemingly unrelated cells differentiating into neurons should lead us to think something like it makes sense that neurogenesis should happen. Most tissues do regenerate, they do have progenitor cells. And given the fact that we see that unrelated cells in these tissues seem to be able to become neurons, we're probably doing a mistake in how we're isolating our progenitor cells. Or at least it should make us say, okay, we'll shut up about the subject until we have better tooling. I can only think of one example, which is to say that, if the brain was muscles in a pre-HRT world, we would conclude something like, oh, once you're like past the age of 60, you can never grow muscle again, because at the macro level, muscle mass decreases. So there must be no such thing as like a muscle growth mechanism that's preserved in 60-year-olds. It just keeps declining. But updating on findings like this is hard because you really have to break out of a lot of other bits of your model in order to update on it. Like findings like these challenge ideas. Oh, cell differentiate across like hierarchies. And these hierarchies of differentiation are like, no. It portrays a lot of fuzziness, where we see that, oh, cells have like a variety of functions in terms of what they can differentiate towards, and often enough, what we think the place in the hierarchy of a cell is actually mistaken. And maybe there's no hierarchies at all, or maybe we misunderstand how these work. It also challenges things like our ability to extract cells from a living organism and actually say anything meaningful about them by looking at them or cultivating them in vitro. So we prefer to just ignore this finding that there are progenitor cells all over the brain and just say, oh no, there's no such thing as adult neoneurogenesis. This is like a finding of sorts that's quite old, but it's, I want to look at one aspect of it, which is blue zones and old mice. So an interesting thing about blue zones is that there's no pattern to them. The GDP per capita is all over the place. The healthcare spending is all over the place. And the genetics are all over the place. Yet all of these zones independently seem to yield among the highest aggregate lifespans and also among the longest lived humans. The other interesting thing is mice, where if you take the common house mice, the oldest lived ones are just wild type mice that were fed a calorically restricted diet, which makes them live for a few hundred days longer than just feeding them chow. So yeah, this should, at least to me, this looks something like this. We have a strong indicator based on these two findings that we have no idea what aging actually looks like. Usually when we figure out a part of nature, we become better at it than anything that nature can produce out of its distribution right out of the box. We figure out the laws of motion, and quite soon we have engines, and we have cars, and we have trains. And those are much better than horses. Well, that's a bit more than the laws of motion. It's laws of motion and also combustion. Or we figure out aerodynamics, and quite soon, even before aerodynamics really work, we have planes. And then we have drones, and soon enough, we have beat some of the abilities of birds, and given a few dozen years, we're essentially better at flight than almost any bird in almost any circumstance. By this standard, we have yet to accomplish anything with aging, really. The only thing we can do when it comes to figuring out how things age and how things age in the slowest possible way is to capture examples from nature and say, look at this, look at that. Oh, slides are not moving. Whoop. Okay, another interesting finding. The fact that vasculature cannot be decoupled from cognition, and this is quite a hairy one, so I won't go too deep into it. The short of this is that there's a lot of recent interest in this link between vasculature and the endothelium of, between cognition and between neurons and dendrites and the endothelium of local blood vessels. And this connection seems to hold and seems to influence cognition at the lowest levels you can look. So in some way, I would say it's arguable that the endothelium is really part of the central nervous system, which is both very interesting and very problematic, given that all the devices we use to study the brain are heavily confounded by endothelial activity and the liquid contents of blood vessels. Another one, which you do know of, is the vibration theory of olfaction. But you know about this for the wrong reasons. You know about it because a pop-sci author made it popular, not because people came up with it 100 years ago. Now, I'm personally very skeptical of this theory, and I would say take no stance whether or not there's any electron tunneling involved in the function of G-protein coupled receptors. But the interesting thing is that our study of this theory went from 0 to 100 in a very rational way, where a pop science author made it popular and then people started trying to prove it or to refute it. It's not that it explained some things that were otherwise unexplained. We had some interesting experiments and we decided to look into it. It was essentially promoted by the media. And then we started talking about it, which seems an awful lot like not how science should work. Finally, a thing that you all know, again, is that exercise remains the only meaningful longevity intervention. That's the only one that anybody that really cares about longevity is doing. And we have no model of biology that explains why exercise works so damn well. More importantly, we don't have a model that tells us what the limits are, both in terms of how much you can do, in terms of the diminishing returns, or anything else. We do have data, we have epidemiological data, but we have no model that tells this to us. We do look at exercise, but we look at it piecemeal through other frameworks, which is quite interesting given that is the only longevity intervention which actually works. Why I'm giving all of these examples, and I don't think any of them are conclusive in any way, they're just indicators. But they're indicators towards this thing that we demand more evidence of things that do not fit our paradigm. Right now, I'd say that all longevity funding is essentially reserved for either reprogramming, small molecules, or biologics. And we have no tentative proof of age reversal in humans coming from any of these approaches, in spite of tremendous scale. Yet the standard issue of, is there a useful therapy, is posed to any other paradigm that wants to make it into aging. So on the one hand, you have one bar, which is we should have a working theoretical model for these paradigms, which we sink hundreds of billions into. And on the other hand, if you want to come up with a novel paradigm at aging, the bar is it should already be able to achieve what these paradigms that we are sinking hundreds of billions into researching are yet unable to achieve, which is rather annoying. One would expect that novel paradigms and novel findings would be treated as interesting and worth researching, even if there's nothing insanely useful about them. So I want to maybe give another example here before I go deeper into this, which is bridges and model of the universe. And I find it quite funny that we think of the universe as this thing that we obviously understand, we have a good model of it. It's a hard fact. It's part of, many people's metaphysics. They will talk about the death and the birth of the universe and stars and galaxies far away and so on and so forth. We see theories about the universe as a very fundamental thing. And we see bridges as just, a human abstraction. Bridges have fuzzy boundaries. There's nothing fundamental about bridges. Yet, if you look at it for time, bridges tend to look an awful lot the same. You can come up with many ideas about how you could cross a body of water, yet we tend to build bridges out of the same materials. We tend to find that we get better at it. We tend to find that bridges built 1,000 years ago or 2,000 years ago still work. I've walked across many of them myself. On the other hand, if we look back at models of the universe, we tend to change these about every 200 years. And whenever we look at the past model of the universe, we think, oh God, how silly that was. They assumed that the sun and the moon were disks. They assumed that everything spun around the earth. They assumed that they were like primordial elements representing the planets. They assumed that galaxies fit into this weird fractal shape, and that the universe had very complex boundaries. But obviously, our current model is correct, and no future model will come in 100 years to challenge that. Another interesting technology I want to look at is grafting, because grafting relates quite closely to biology, and especially to recent advances in understanding genetics and understanding the interactions between tissues. Yet, if we look at grafting, we are doing the same thing. Grafting has improved a lot, but the fundamental concepts, the fundamental concepts have remained the same, such that if you take a druid from 3,000 years ago, they would likely be able to execute, if not understand, modern grafting practices, given a few months of training. I lost my notes, so allow me a 10-second break here, which we can cut from the panel recording.
No worries, we have time.
So with all of this preamble, you might think this sounds somewhat motivated. You are setting out a narrative here, and you are correct. Because I do want to talk about my own findings, or rather, not really about what they involve, but rather about how they are received, which I found to be quite interesting. So if I am to blow my own horn here in a way that's maybe a bit undeserved, I would say that I managed to reverse cognitive decline by about a decade. I had the simple thought that one of the things that you notice with the brain is that it decreases with size as one ages. And with almost any other tissue, the one thing that seems to increase the size of it is if you just increase perfusion. Additionally, if you increase perfusion often enough, you also lead to increases in vascularization, which should make that change permanent, and it should make it have lasting effects and maybe even reinforce itself once you're done with the intervention. Therefore, if I can safely increase the perfusion of the brain over a long enough period of time, maybe we can reverse brain aging. Given the fact that I'm one guy, I have a house and a few friends, I can only propose very simple ways for people to do this with methods that are already well known and combine them in ways that make sense. So I designed a relatively safe intervention that did something like increase perfusion and have people go through challenging but not difficult motor tasks of the kind that might require one to recruit or create new pathways, like, learn how to lift your middle toe. And I recruited controls, and I had a very rigorous protocol for an RCT where my controls got plenty of advantages over the interventions. And I observed significant changes in my only endpoint, which I took as a very hard to fake endpoint, fluid intelligence, measured across multiple tests. So I was ready to find out what's happening. My questions were of the kind, how does increased perfusion regenerate the brain? Does this finding hold if I scale it up to 100 people or to 1,000? I only had 17. How could I improve this protocol? More importantly, what's the underlying mechanism? What is the vasculature doing during this? Is my theory even correct, or does this have nothing to do with perfusion? What are the potential confounders here? Can I observe improvements in other markers that relate to aging? These are all questions I wanted to ask given these promising initial results. But instead, what happened is that my data got promptly ignored. And it didn't get ignored in a way where I didn't have the reputation, or in a way where people ran their own independent analysis and said, hey, I don't think this is actually statistically significant, or I don't think that 4 points of increase compared to control is that interesting. But it got ignored with a question that sounded like, how does this fit? So everybody seemed to have the same complaint. I'm not selling a drug, nor am I designing a drug. So this is not medicine. I'm not doing something at the level of genes or at the level of cells. So I'm obviously not doing biology. I'm not using anything like an epigenetic clock. So I'm not doing aging. So when I was hoping for productive pushback, the scientific kind, that's asking me, hey, what happened here? What are the confounders? What would be follow-up experiments? What would you need to do them? Instead, I really got, your data here doesn't really fit into any model. So yeah, you did manage to improve maybe this one very important marker that everybody says is very hard to increase and shrinks with aging and seems to be potentially the most relevant thing for a functioning human in today's society. But this doesn't quite fit. So this is really the thing that motivated this talk and has motivated the last few months for me for really looking at what are other overlooked findings that are maybe unpublished or maybe got very little attention, even though they prove things that we collectively think are impossible, just because they don't fit into any ontology, they don't fit into any theory, they don't fit into any narrative. And one thing I want to say here is, what does it mean to take an unusual finding seriously? And I think the best way to actually phrase this is by saying, what does it mean to by saying what taking an unusual finding seriously doesn't mean. It doesn't mean trying to sell it as a therapy. It doesn't mean trying to make it sound more scientific. It doesn't mean trying to reduce them to diminish what you're doing until the effect is minimized. It doesn't mean to try to get insurance to pay for something resulting from it. And it doesn't mean trying to reduce it to a pill or a device that can be FDA approved. What it does mean is asking, why is this finding fascinating, given our model? And what is the underlying process here? Why couldn't we derive this finding, given our existing model of biology? Why is this surprising? I will go back to another point, just to again, link this to why is this relevant for aging? We don't really understand aging. And we build this understanding by looking and then by doing. We can only fit observations. We can only fit the results of what we've done into models. And currently, when our models fail to solve aging, we look for more observations that fit the models in order to throw at the models. We say, the problem is complex, we just need more data. Instead, I think we should, in some way, look differently. We have to have an early barrier of, does this actually work? Is there anything unusual related to how an organism ages here? And if that can be proven, then I believe we should be more open to relaxing our models in order to integrate new data. And this is not to say that we should create a new Manhattan project based on this or that quark theory of aging, because there's like 1,000 of them. But maybe all it actually takes to solve aging is something like $10 million spent behind the correct approach. And adjusting for 2024 figures, airplane cost us 34,000. Fission, at a relatively high estimate, cost us 200,000 to figure out. The first transistor, hard to find out, probably less than 2 million. Falcon 1, 100 million, quite a lot, but still much less than one would expect given existing efforts. So what does it look like to actually, may I draw for a second? So what does it look like to fund these sort of like 1 million experiments to solve aging? It looks like demanding a transistor, but not demanding the computer. It looks like not demanding a Boeing airplane, but demanding a flying bicycle. So I think in order to fund the kind of experiments that will at least have some hope of solving aging, we have to ask for things like functionally immortal rats that are able to fight, run, swim, and breed at age 20, as opposed to dying at 4. We have to ask for a 100-year-old human beating a 100-meter speed record. We have to ask for a 40-year-old getting the same antibody titer to a vaccine as a 5-year-old. We have to ask for 30-year-olds being able to regrow a fingertip, or for 25-year-olds doubling their amount of gray matter, which probably would involve changing the morphology of the skull to some extent, or for a 20-year-old to grow a tail. And these all seem crazy, but there's nothing to say that these findings are hard, that they require Manhattan Project level funding to achieve. They do probably require looking in some other place than where we're looking right now. What we shouldn't do is demand FDA-approved therapies because we have about 20K and none of them solve aging or put a dent in it. We shouldn't demand citations because we have, about 100 billion collective citations flowing around for the last 100 and something years. Maximum lifespan has been unimpressed. Fancy words, they don't really seem to solve aging. There's a lot of complex theories out there. They don't seem to put a dent in lifespan. So essentially what I want to invite here is that we are roughly in a pre-theoretical world when it comes to aging. And what we should really be looking for are interesting findings of the sort that prove to us that maybe there is some hope to reverse the process of aging, that maybe there is a way to increase maximal lifespan other than just looking in nature and finding the organisms that are longest lived. The final message that I basically have for any independent researcher here, and I know there's at least 5 of you, is, don't give up. Doing research that doesn't fit a paradigm seems to be hard. Convincing people to think differently is basically impossible, and you have to meet them 99% of the way. And I think that's a fun challenge. The chance of an idea being wrong is not very relevant. It should be weighted against the potential impact if that idea is correct. And overall, you're allowed to be wrong because no model works right now. So at most, you're just average. That's about it. Any questions?
Thank you, George. You get a thumbs up at least. Let's see about the questions here. That was very entertaining, building up on many of our kitchen conversations. So let's see who we have as questions here. Feel free to raise your hand. And also, if you have a question in the chat, I will prioritize those for now. So Steve, you had a lot of them in the chat already. If you want to go ahead.
Those weren't really questions. I was discussing different points and stuff, but it has been my observation that the institutions of science restrict the box to narrow paradigms, and by that process exclude stuff that turns out to be more, not only more interesting, as you're suggesting, but also more productive in terms of delivering value. So I applaud your approach, and I've spent my whole life digging into that kind of information.
Yes, thank you. Very good presentation. Definitely loved your mindset, and see a lot of overlap with how I think about the problem. Would love to be in touch if you could drop your email in the chat. And one of my questions was, you were talking about a process you went through that looked like it did have some good results. And even though it didn't fit a current model, I was wondering what intervention you were doing there. And it sounded like it worked, sounded pretty good. So I was just curious.
Yeah, I think it worked, but it, generally speaking, takes like 2 weeks and 4 hours. So it's quite hard to get. I actually, other than my friends, which I peer pressured a lot into doing it, so I had one dropout. I gave the data of how to do this to about 30 different people that were, I selected them actually. Like, people send me emails, and then I was like, okay, who's serious? Contact me on Signal, have a 20-minute chat with me. And then nobody out of those 30 people really pulled through with it. But I will draw my email, and you can just email me and I'll send those notes. And I do plan to publish this as well. So I'll drop the link where this will be published. I'm currently waiting on longitudinal data so that I have like the results a few months after, as opposed to just the week after, before I publish everything. But yeah, it's fairly boring. It's the kind of finding where I'm like, it doesn't, like, in itself, it's something that intuitively seems like it should work. You just take the kind of drugs and do the kind of like inversions that increase perfusion and to some extent motor confusion. Really, my interest was, and to some extent still is, what's actually happening there, and can we in some way build a theory and capture that process. But sadly enough, that's significantly more challenging than just doing the thing.
Makes sense. Thanks. I'll send you an email, and we could go from there.
Yeah, I've dropped my email, and I will drop a link to where I'm publishing some of these things.
Cool. Thanks.
Yeah, hi, George. I thought it was an interesting talk and everything. And I think you're right that if you get more perfusion through the brain, it's probably a good thing. And there are some drugs that do that. Some of the PDE5 inhibitors have been claimed to do that. Even the multivitamins, which may have niacin. Niacin has other problems, cardiovascular stuff, but it does probably increase perfusion through the brain and things like that. So I think that's true. I think one of the big problems is, running a lot of trials in parallel. I think Steve mentioned something about the FDA-approved drugs, and he could be partially right there. And none of the things were run in parallel. And there were efforts to try to do comparative efficacy trials many years ago, even on approved drugs, and Congress really shut them down because most of the drug companies just like to compete in advertising, not in actually comparative efficacy. And, so I think those are some of the problems that they found. It's much easier to run advertisements to get people to use their drugs to actually show which one actually works better and things like that. And they spend much more money on it than they do on research. But those are issues that are industry wide and not just isolated to the FDA. The FDA is, we just have to approve what's put in front of us. And, sometimes, it's lacking, but you have to make a decision to go one way or the other. And I agree that we're going to probably waste billions of dollars on things that are, not giving you that much. And, but those are always issues. Yeah. But I think it's great setting up things where you can do parallel trials to show what is better than each other. I think is the way to go. And for cognitive functions and studies, I think that's a great way to do it.
Yeah, I think so. Generally speaking, I'm always pro more citizen science, more parallel trials. I even worked the company focused on this problem for about 2 years. I do think there's something to be said, at least about my particular pet peeves, not really being related to this. My current gut instinct is that we could actually be running trials 100 times faster. And I think this would be great. And I think it would do great things for median lifespan. But really, the thing that I think we're failing is to actually ask, how do all of these things work? Look at that process of regeneration more closely through a lens that actually allows us to improve it. And I guess that's the thing that I would really like to see. I would like to see the kind of substances or combinations that lead to systemic de-aging be studied as far as the mechanism goes. I do think we could essentially be doing a lot more trials, a lot more cheaply, to figure out what the best combination is. But I think even the best combination doesn't solve aging.
A lot of this stuff is cellular differentiation. And when you go to de-differentiation, you end up with potential really big problems. And I think those are some of the issues that if you start creating a lot of stem cells in the brain, are they going to evolve into, cancers, glioma, multiform? Is that you see a lot of different types, but I think that's one of the big fears. And, if you go all the way back, you're basically doing de-differentiation. Differentiation, has somewhat of a program associated with it. And unfortunately, that's also probably an aging program.
Yeah, so my limited take on that is that I do think there are mechanisms which seem to have similar effects, but without the cancer is seemingly localized expression of hypoxia related proteins seems to often work. Literal, like physical manipulation that leads to increases in perfusion seems to also lead to some de-differentiation, which for some reason is like non-cancerous. Even injury, which in certain types of tissue could lead to quite a lot of the de-differentiation, doesn't seem to be very correlated with cancer. But, I don't have a perfect picture of this. I do agree that most therapies that do reverse aging have some of that risk associated with them. I do see a lot of red sands. So
maybe we'll just move along for now, and maybe we can get back to it with more time. Mark, you go next, please.
Sure. So firstly, I'll say that I strongly agree that there should be more support for out-of-the-box ideas, and I've actually had a lot of people come to me with ones that they can't get support for, and I just like figuring out a funding mechanism for that would be amazing because there really aren't good ones. But what I've been in the industry for a long time, and I think of it as I think we maybe are in the worst case scenario where we actually do have a model. It's just that the model tells us that we're way, way at a too primitive a technology level to do anything about aging, and that there is actually no easy fix. There is no silver bullet, and that aging is just a matter of a network of homeostatic metabolic pathways, thousands of them, that are all accumulating deviations and all affecting each other and going exponentially off the rails, and that there's no single point of leverage that you can push on that will have a big effect because everything is actually failing. And so the only way you could solve aging would be to have some sort of technology level that is way beyond what we have now in terms of engineering when it comes to biology, or a replacement type approach, John Haverhill would. So that's yeah. Do you think that worst case scenario do you worry that might actually be the case, or are you more leaning towards there is some kind of high leverage point? And if you think there's a high leverage point, what makes you think that there is one?
yeah, so I have two separate answers to that. The brief answer to that is my intuition tells me no, we might not be in a worst case scenario. And really the fact that we haven't put a dent in aging thus far yields me this intuition, in that the vast majority of cases, like even silly things like people trying to fly and using like fake bird wings, when people actually just try to do a thing that at least tracks their objectives, as long as they've actually understood the problem even a little bit, we seem to be able to progress a little. And then often we hit a hard wall. Right now, my impression is that we have actually made zero progress, which tells me we might just not really understand the problem. And thus, I don't think we've hit a hard wall yet, if any exists. That's pretty metaphorical, and I think that's contentious. That's an intuition. I think the more realistic answer to that, which really is the answer that directs my own research interest, is that, yes, the problem might be complex enough that it's almost impossible to solve. In that case, we should be tracking the fact that we should try to solve it for the bits that are relevant. In my model right now, that is really the CNS. If I asked you, would you be okay with all of your nerves and neurons being placed in an artificial body? I think most people would be like, yeah, that's probably preferable to even being 60, let alone to not existing. So I think focusing on that alone is the correct approach in the hard problem case, because anencephalic clones don't seem to be that hard. I would further say that if this is indeed a complex control problem, the one computer that we know has control over this complex mechanism is our very brain. Because our brain seems to be able, well, not necessarily the brain, but the CNS, and really even the peripheral nervous system, it does seem to be able to regulate a lot of these complex processes very well. To the extent that some of the things we do are at, on the nanosecond or even millisecond level, we're able to establish feedback loops that involve hundreds of proteins having changes in their expression. And these changes are, primarily fueled by cognition as opposed to anything else, which seem to lead to downstream changes. So I think if you track this as a complex problem, you should be thinking about how to offload as much of both the data gathering and of the processing on things that are or are very similar to nervous systems. Though that, using ex vivo nervous systems might have other ethical implications. Personally, I tend to focus on what's possible within the one that is integrated within us. So hopefully that answers it and I haven't wiggled away from the question.
That's yeah, I hope you're right. My suspicion is that we are in the worst case scenario.
Hopefully we can get our way out of it. Marina, you can next.
Yeah, thank you. I don't know if I will be able to formulate a question in the end, but I have a ton of different emotions after your talk. So, like, on one hand, the study design with 17 participants is not even marginally enough to have a statistical significance. I don't know if you're using P-level statistics. So that's one point. Maybe use some other methods. That's another point. Maybe you're approaching it differently. So I guess you just got a bad reviewer who was asking you to fit the paradigm. The reviewer should have asked you, what is the new paradigm? Like, having spent over 10 years in neuroscience, I can tell you that there is a 95% probability that your idea or finding is wrong. I took it very personally in the beginning because I had null findings all the time and I thought I was doing something wrong. And then I realized, observing others, that everyone has those null findings all the time, and that there is a very little chance to get something really valuable if you are relying on the previous knowledge. That's the thing. And now I am in Cerebrum DAO where we are looking for new ways to treat Alzheimer's. And it's very difficult to find something new. And it's just indeed this state of the art that we don't know how to treat it. We don't know how exactly the disease develops. And yes, the drugs are bad just because the current state of knowledge is bad. But how do you find the new treatments which, one, will be working? Second, which you could actually implement in the healthcare systems is a huge challenge. Yeah, I guess I'm just commenting. I really liked in the end that you had a measurable endpoint with 60-year-olds, 40-year-olds. Not all of them, though, make sense biologically. Maybe the antibody titer, similar to young persons, makes sense from the medical point of view, but regrowing the tail, I'm not sure it's the right endpoint to track. Yeah, so I guess you're still very young and ambitious. It's really cool. I'm looking forward to see how you develop. And with your current study, just the only way to prove that you're right is to show evidence. So try to find a follow-up study, I don't know, funding for it, or do it along your other work. Yeah, just commenting. But lots of emotions.
Yes.
Thank you.
I could give a few answers there. I'm not exactly sure about your statistical significance comment there. I actually just ran the analysis now to make sure. Essentially, using related t-tests on Raven's IQ, Raven's progressive matrices, which is the fluid intelligence test that I think is most well regarded. I used 4 of them and I had significant findings on all of them. I'm just going to code ravens and on the combination of them. And I used a crystallized intelligence test where I had no findings as a control. But anyway, so the p-value that I got on the intervention group is 0.006 on a 6-point change, as opposed to the control group, where the p-value is 0.2. So not really significant, but there's some learning, which means there's like a 0.7 increase. And the p-value that I got when I compared the changes with an independent t-test, so compare the changes in the control group with the ones in the intervention group, that's also significant at 0.025. So I do think I got significance. And also, to my knowledge, I got the highest increase in fluid intelligence that has ever been reported. Though I essentially welcome data criticism, which is why I gave people both the analysis code and the data whenever asked. I don't think anybody had an issue with this ever at the level of, oh, your data analysis is wrong. On that front, I think this is essentially my specialty. I primarily did research in machine learning and to some extent statistics. I could see a lot of criticism with how I could have had my methodology be even better. I guess the interesting thing for me was nobody really had a criticism at that issue. Most of the things were at the vibes level, at the, oh, this doesn't quite fit the pattern of what it looks like to do science. So you should try to change the vibe a tiny bit. Yeah, I think another interesting point there that you bring up, which I certainly see as being somewhat true, is this idea that it takes time. And I basically agree with. I've come to accept that this is a long process. I'm still somewhat in the bucket that I think if we look at the thing historically, time is not guaranteed to solve the issues. And I can only put myself in the shoes of, 10,000 years of scholars throughout humanity and really think that for 99.9% of them, it's not only that they didn't really produce something that was relevant, but they never really stood a chance. They were so far from an ontology that could have been productive that you could create, a million St. Augustines. You can create a billion St. Augustines and have the Manhattan Project for, studying idealist Catholicism and how that derives the laws of nature. And you get not an inch closer to understanding fission or having computers. And then when someone seems to stumble upon something, you really only need 4 or 5 physicists to put the basis for what is most of modern physics over 100 years. So yeah, I do agree this is a long problem, but I guess my intuition still remains that it's more of a step function. And you really have to do the kind of digging that allows you to look for a step function, as opposed to hoping that you can do anything incrementally. I'm a fan of incrementalism as an engineer, but it does not seem to work in science yet.
All right. Thank you so much, you guys. We have to gradually wrap it up here. Do you have any final words, George? Any outlook, any ways that you want to collaborate with people in the last minute before we close it down?
I have my email in the chat, so anybody that's interested in this in any way, do feel free to email me. Right now, I am doing my research and I'm trying to fund one independent scholar to do the research with me. Given that I'm self-funding all of my experiments, I have only limited funding. So any volunteer that wants to lend me expertise, especially someone that's interested in complex biomedical devices, which I'm currently trying to build from scratch, or has various of these devices to lend, I would be quite interested. But anybody with weird yet seemingly practical takes on neuroscience, and really anyone that feels oh, this fits their pattern, I would love to connect.
Awesome. Thank you so much. Thanks everyone for your great questions. Really enjoyed this one. Have a good one, everyone.